Great. So, welcome to the BioEng seminar. This week, we have two of the newest additions to the department, two new group leaders giving talk. And the first speaker is Dr. Nuria Oliva Jorge, or Jorge Oliva, sorry. No?
Oliva Jorge.
Oliva Jorge. She is the brand new Imperial College Research Fellow in the department. I guess you started this fall, this academic year, is that correct? As the fellow, but she has been in the department for a bit longer. She is an organic chemist by training, earning her PhD in 2016 from Harvard MIT Division of Health Science and Technology.
Naomi seems to have become muted, sorry.
Yeah, no problem. Yeah, okay. And so you got PhD from Harvard MIT Division of Health Science and Technology, working on bioadhesives for wound closure and the delivery of nanotechnology. You've been in the area ever since, maybe? That it rings a bell to me about your research. But and then a postdoc fellow in Boston, stayed in Boston at Harvard Medical School with Brigham Women's Hospital before hopping over the pond with a Marie Curie action postdoctoral fellowship at Imperial College London and Institut Clinique de Celia. I think I just messed that up in Barcelona. So yeah, and Nuria, so yeah, I have this tradition of sharing something about the speakers that's not usually on their scientific CV, and Nuria is a Zumba dancer and also squash player. So if you're looking for well, if you want to join her, I guess you should get in touch with her. And so yeah, with that, I would like to hand over the mic to Nuria, please. The floor is yours.
Great. Let me share the screen. Okay, so can you all see the presentation now?
Yes, we can.
Yes, okay, wonderful.
Yeah.
Okay, yeah, so Naomi, thank you very much for the introduction. Yes, I do like Zumba and squash, but my enthusiasm is far exceeds my skill, so don't get too excited about any of those hobbies. And I'd like to thank the department too for inviting me to give this talk. And before I get into the science per se, I want to give a little bit of a, because I'm fairly new in the department and some of you may not even have heard of me before, just give a little bit of a background, and Naomi has covered most of it. Sorry for all the foreigner names, apologize for that. But yeah, like Naomi said, I'm an organic chemist by training with a minor in chemical engineering and graduated back in 2009. And I started doing a bit of biomaterials in my undergraduate, and I really loved them. So I took the opportunity to do a research experience at MIT. And I loved it so much that I actually stayed for a PhD in medical engineering and medical physics, where I developed medical technologies based mostly on adhesive hydrogels to deliver chemotherapy to tumors and also to promote wound closure. And I graduated in 2016. I stayed for a little longer to do a short postdoc. And then in 2018, I got a fellowship to join the AMQUIS lab in the department and do my postdoc on developing wound dressings for treating chronic wounds, which is what I'm going to tell you about today. And I also, in parallel, I worked on, I worked with Ben on his strap technology to heal broken bones. And as of last September, I started my own group. I moved to White City, and I'm an Imperial College Research Fellow now, also in the department. And so my group sort of lies at the intersection of biomaterials, medicine, and biology. And what we're trying to do is we're trying to develop medical technologies based mostly on hydrogels. Let me just do the pointer. Based on medical hydrogels and nanotechnology often combined with each other to tackle human disease. And I'm also very interested in understanding how biomaterials interact with tissues at different levels to better understand disease progression and how we can use biomaterials to alter disease. And today I'm going to tell you about one of the projects, as I said, the one that I started as a postdoc fellow, and I brought to my lab with me at the intersection of hydrogel technology and nanotechnology to heal chronic wounds. And we do so by delivering gene therapy. And this is something that's new, that hasn't been done before. Gene therapy is not new. The use of nanotechnology to deliver it is not, but its use to chronic wounds and wound healing is actually something that, as I said, hasn't been done before. And chronic wounds are actually, they're wounds that they won't heal. They're mostly ulcers, and they can take weeks to months to heal, and they affect over 1.2 million people in the UK every year. And the very alarming piece of information is actually that there has been an increase of 93% in their incidence between 2013 and 2018. And the main reason for this is actually the aging society and lifestyle choices. And as of 2018, the NHS actually spent about £5.8 billion managing these types of wounds. And the way that they're managed is often with wound dressings. And over the last decade, there have been a number of what's called advanced wound dressings, sometimes antimicrobial dressings that aim to accelerate the healing of these chronic wounds. And they're also a lot more expensive than normal dressings. But actually, the last evidence summary from the National Institute for Health and Care Excellence revealed that there is not enough evidence that any of these advanced dressings or antimicrobial dressings have any effect on healing. And this is sort of the unmet clinical need that we decided we wanted to tackle as part of my postdoc fellowship. And there are, for that we need to take a step back and try to understand why do these wounds don't close. And one of the main reasons, there are many different reasons, they're very complex environments, but one of the main reasons that's widely accepted now is that chronic wounds have an altered gene expression profile in chronic wounds. And so this is one of the studies. There are many, many studies that show this exact same pattern, but this is actually very clear, where they took cells from the edge of a chronic wound and cells that were a little bit farther away. And the first thing that they saw is they look very different than to normal cells. And they also have a very hard time migrating to closest scratches, which is the equivalent of like a wound on a plate. And when they looked at the expression profile, they realized that actually there's there are many, many genes that are either down or up regulated compared to healthy cells. And when they took the top genes that were dysregulated and they gave these genes as part of sort of gene therapy to those cells in location A, they start to heal similar to cells in the control or location B. So this suggests that actually gene therapy is a pretty good treatment option to restore healthy gene expression. So why haven't we done it yet, right? So doing gene expression in fibroblasts is actually something that's not new either. It has been done. So there are studies dating back to the 90s where they take fibroblasts and they genetically modify them with different genes, but they always do so ex vivo. And that means outside of the body. That means that you take cells from a patient, you grow them in a plate, and then you transfect them with the genes of interest, either using viruses or liposomes. And then you detach them from the plate, grow them in a scaffold, and put them back in the patient. And this can take weeks to months. And of course, it's time consuming, it's expensive, it requires a lot of very specific infrastructure. And there's always a high risk of infection in the patient when you're implanting foreign materials back into a person. So the more ideal scenario is actually doing this in vivo, so directly transfecting fibroblasts in the patient. And the way you can do that is either the same approach, right? You can use viruses, you can use liposomes, you could potentially use nanoparticles. Viruses and liposomes have been used clinically. In fact, most of our COVID vaccines are liposomes loaded with messenger RNA. So it is possible, and they all have their advantages and disadvantages. And as far as I know, no gene therapy at the moment is being done with polymeric nanoparticles. But then, as I said, they have their advantages and disadvantages. And of course, the main disadvantage of a virus is the ability that it has to trigger an immune response and off-target effects, which doesn't happen with liposomes or polyplexes, or not to the same extent. But of course, they cannot protect the genetic cargo and transfect as well as the virus would, which that's what it's evolved to do. Now, the main advantage of polyplexes or polymeric nanoparticles that I want to highlight here is that the possibility of incorporating them into scaffolds for local and sustained delivery over time. And why is this important? Well, if you think of a vaccine, you just get that intravenously, right? And it goes everywhere, and that's perfectly fine. But if you think of tissue regeneration or wound healing, you actually ideally would want to deliver those nanoparticles at the wound site, so locally. And not just that, the healing process is a process that takes time. So ideally, you would want to deliver those nanoparticles over a therapeutically relevant window. So it could be a few days to a couple of weeks. So taking all of this into account, then we proposed, as part of my postdoc fellowship, to develop this cross-linked wound dressing that was injectable, so that can fill an irregular wound, which is very common in chronic wounds. It would cross-link at the wound site and then deliver nanoparticles that were loaded with gene therapy that can then enter fibroblasts and sort of restore the gene expression of healthy fibroblasts, so that we can promote healing in the wound site. And for that, we decided to join forces. So join my expertise on injectable hydrogels with Professor Roses from IQS expertise in polyplexes for gene therapy, and also Ben in the department, who's an expert in wound healing and the biology behind chronic wounds, and in particular, diabetic foot ulcers. And we also brought Tony on board, who is a PhD student from IQS, who is now actually doing his final year PhD here at Imperial with me with an Erasmus scholarship. And we've recently submitted this paper. It's under a second round of revisions, but it's actually, if you're interested in the details, it is publicly available in the Chem archive. So we started by developing the nanoparticles, and these nanoparticles are based, and forget about the chemistry behind it. It's just like a core polymer that's made of polyvet amino esters, or PBAs. And these are positively charged polymers that, when they encounter this negatively charged genetic material, forms discrete nanoparticles through electrostatic interactions. And when they enter the cells to an endosome and the pH lowers, these amino esters actually get degraded much faster, releasing the genetic cargo inside the cells. Now, what's also very interesting is that they can be conjugated at the end with these oligopeptides, so short peptide sequences that what they do is they enhance cellular uptake and endosomal escape, so they enhance transfection efficiency. And different cell types require different polypeptides or oligopeptides or combinations of those. So this is from a paper by, like 7 years ago, but there are like tens of papers from Professor Burrosa's lab in Barcelona, where they've looked at many different types of cells, mostly cancer cells. And actually, they're recently working on another COVID vaccine using these polyplexes. But one common theme, if you look at this HNDF, which is human neonatal dermal fibroblasts, the expression, the transfection efficiency is consistently low in all of the with all the formulations, and this is a common theme. Cancer cells are actually very easy to transfect. Healthy cells, not so much, and fibroblasts are actually fairly difficult to transfect with nanoparticles. So this was the first problem or challenge that we wanted to tackle with this project. But also the other advantage of using these nanoparticles, as I mentioned, is that we can incorporate them into scaffolds for delivery. And we did this actually a few years back. We incorporated these nanoparticles as a collaboration in the adhesive hydrogel that I developed during my PhD, and implanted them in mice bearing luciferase expressing breast cancer. And we gave them nanoparticles to silence luciferase. And what we observed is that we could actually, we had sustained silencing of luciferase over time. So we have a platform that we can tune for different cell types, and we can actually make it translatable to a product that can be used in vivo. So Tony took on this challenge to figure out how to transfect fibroblasts, and he did an amazing job. He tested many different formulations, and in the interest of time, I'm going to go directly to the optimal, which is 6RH. But in general, he altered the hydrophilicity of the backbone and also the combination of many different oligopeptides, and eventually found that this C6, which is a hydrophobic backbone, with arginine and histidine in a ratio 2:3, led to nanoparticles of about 127 nanometers in diameter that were highly cytocompatible, highest cytocompatibility compared to any other that we tried, and also had very good transfection of dermal fibroblasts, not just in terms of the number of cells transfected, which is almost 80% compared to, this is the commercially available control that transfects about 50% of fibroblasts, but also you can already see here from the intensity of the signal that we managed to transfect much more, so hence much more fluorescence being emitted, and also this peak is a lot sharper, so there's a lot more consistency in the transfection. So we've got a really good candidate instead in terms of like nanoparticle formulation, and now we need to incorporate it into a wound dressing for delivery at the wound site. And we decided to make a new injectable hydrogel that was based on the same PBAE backbone, so the same polymer as we use for the nanoparticles, because we hypothesized that actually using the same polymer would enhance the stability of our particles. And in order, we made it a little bit more hydrophilic to make it more like a hydrogel structure. But not just that, we also, in order to make it cross-link, we conjugated this thiopyridyl group that reacts spontaneously with thiol groups. So when we mix these PBAEs with forearm peg thiol, we form hydrogels. And you can see here, this is like the hydrogel structure and the microscopy images. So we tested two formulations, which we call 1.1 and 1.4. And 1.1, the numbers are basically the PEG to PBAE ratio. So this one, there's equal molar ratios of PEG and PBAE, while this one has 4 times more PBAE than PEG. And the rationale behind this is that by increasing the concentration of PBAE, we wanted to increase the cross-linking density, which would in turn reduce the pore size. And all of this together would lead to a longer degradation time and also longer release profile of nanoparticles. So we wanted to have control over that release. That being said, if you look at the structure, you can actually see, yes, there are areas with much smaller pores in general, but there are also very large pores that can be seen. And while the average pore size, you can see here, this orange line is the same, the mean pore size actually is smaller for 1.4 than it is for 1.1, which aligns with our hypothesis. When we look at
I'll mute it.
Sorry? Is there any problem?
I think somebody's mic was on, but
okay.
it's been muted. Yeah, yes, yeah.
Okay, that was okay. No worries.
Yeah.
Yeah. So when we looked at the whole, because these are sections, so this is only 2D. And when we look at the whole structure through SEM, we actually saw that those larger pores were in fact cracks or fractures. And this is never a good sign, because fractures are actually points of weakness in the material. And this is exactly what we saw when we did mechanical testing. Even though they're both viscoelastic materials, and yes, the 1.4 has higher cross-linking density, which means the elastic modulus is 10 times higher. So the scales are not the same. So this is about 10 times higher elastic modulus than 1.1. This material actually broke very easily when applying strain. And when we look at the degradation profile, again, in line with our hypothesis that 1,4 would degrade over a longer period of time, with the caveat that after a few hours, the material started to break because of all those larger cracks. And then again, once you have a material that breaks, then the degradation can alter very much based on where it breaks from. So, and in terms of the 1,1, it no toxicity at any time point. The hydrogel 1,4 actually had a lot of toxicity at 24 hours that we correlated to a burst release of unreacted PVA. You can see if you just add PVA to the media, it actually kills the cells. So all this information together made us decide that hydrogel 1,1 was a better wound dressing candidate to incorporate our polyplexes. That being said, we incorporate them into both hydrogels, and you can read all about it in the paper that's in the Chem archive. But in the interest of time, I'm going to focus now on the 1,1 and how we incorporated the polyplexes there. And the first thing we observed is that incorporating PVA polyplexes actually led to larger pores too, which is interesting. This kind of let us, suggests that polyplexes are interacting with the material, which is good. This is exactly what we want. And while the average pore size increases owing to those larger pores, the mean stays the same, and so does the toxicity, which is close to nothing, actually, compared to untreated controls. And what is very promising is that the commercially available nanoparticles in our hydrogel lead to 40% decrease in cell viability. So they're actually more toxic than our particles, which is good news. We've got a better platform. The other really good news is that actually, because these PVAs are interacting with the whole scaffold and the way the material is formed, we have a similar profile, a little bit longer, about 10 days instead of 8. But actually, this 70% like burst release that we observed got decreased to like about 10%. So this is really good news. And then the rest of the profile resemble almost like a phase 0 degradation. And then finally, we wanted to see, what happens to the nanoparticles. About 50% gets released in a burst at the first 24 hours, but the remaining 50% actually is released in a sustained manner over 9 days. And we wanted to say, okay, is this stable or not? Are these nanoparticles stable inside the material? And at the moment that we formed the hydrogel, we did imaging where you see here that the polymer scaffold, or the polymer shell, and the DNA co-localize, meaning that they're most likely forming nanoparticles, and they're also homogeneously distributed throughout the material. This is a 3D rendering in like microscopy inside the hydrogel scaffold. But it was more important to know whether these particles were stable upon release from the hydrogel, right? Because that is when they're going to transfect the cells. And so what we did is we actually ran agarose gels, and the nanoparticles are actually too big to enter the gel, so they stay in the well, while free DNA runs through the gel. So if these particles are being degraded, then the DNA leaks out, and then you can see it running through the gel. And you can very clearly see that soluble nanoparticles in media, under the same conditions as the ones released from the hydrogel and at the same time points, start to leak DNA at 3 hours, but it's very obvious at 24 and 48 hours, while the nanoparticles that are released from our hydrogel remain stable over time. There's no leakage of DNA. So we have a hydrogel or a wound dressing that can release nanoparticles in a sustained manner over 10 days and actually protect them from degradation. So the last question to answer was, does this actually transfect? And the short answer is yes. We're still tweaking the system, but yes, we can transfect. You can see emission of green fluorescent protein, which is the messenger RNA that we're using to transfect these cells. And what's also very promising is that the commercially available nanoparticles released from our hydrogel do not transfect at all compared to untreated control. So overall, we've got a wound dressing technology for treating chronic wounds with gene therapy that is injectable, cytocompatible, and biodegradable, and that allows for the sustained release of polyplexes carrying gene therapy and high transfection of dermal fibroblasts. So what's next? So one of the projects that is currently undergoing in the lab with funding from the British Skin Foundation and in collaboration with Ben is looking at therapeutic microRNAs for treatment of diabetic foot ulcers, where we're taking some very interesting microRNAs that Mara identified during her PhD, and we're delivering them in diabetic fibroblasts. And in parallel, we're also trying to develop more like translational models that are not applied onto these. So we're, rather than going directly to animals, we're trying to do skin models, 3D skin models that resemble sort of the dermis with all the fibroblasts. And this is, as of last week, we got the first imaging, actually. This is very recent data, where you can see the fibroblasts sitting on the 3D scaffold, actually like interacting with each other. This is really cool. So we're beginning to do a lot of experiments on 3D. And this is something that my Europe, Anna and Tony are doing together. And I just want to use the last few seconds to, for those of you who don't know me, or if there's any students attending this talk and they're interested in any of the projects that we might be having, you can have a chat with me later. But I just wanted to talk about a couple of projects that have recently started, so there's not much going on yet, but they're really exciting. One of them is development of hydrogels to regenerate cartilage that we recently got funding from the Rose-Lewis Trust. My ICRF, which looks more at the interactions between biomaterials and tissue, in particular adhesive and tissues, to try to understand disease and more inflammation mostly, and try to use materials to affect disease progression. And finally, I have a few projects on nanotechnology for treating metastatic cancer. So with that, I'd like to thank my advisors and my mentors over the years, and my collaborators, and of course, particularly Salvador and Ben, who have been amazing with this project and my whole transition to independence and the funding sources. And of course, if you have any questions, I'm happy to answer them now, but you can always send me an email later. Thank you.
Thank you, Nuria. Great talk, past, present, and future. Ooh, lots of hands. Yes, if you have any question, please pop it in the chat box or raise your hand. I might start with the first question. So your hydrogel, do they dry when they are on the skin surface? And then when they dry, does that change how it interacts with those nanoparticles? And yeah.
Well, that's a very good question. At the moment, we're testing them in a well plate, and they're covered by media. So obviously they don't dry because they're always covered. But on skin, they will definitely. So the good thing about hydrogels is that they actually absorb a lot of the exudates, and in particular in wounds, there are a lot of exudates. So once you have the exudates from one end, and ideally you would cover that with like a plaster or something on top, then you would sort of maintain some level of humidity that ideally you don't want it to dry.
Yes. Do you envisage something like Band-Aid, basically some covering to prevent that?
So yeah, that's a great question. I mean, we can make it in the shape of a Band-Aid if we want to. It would have actually been a lot easier because you can do whatever you want on a Band-Aid. You can do UV cross-linking, you can do other types of reaction, but we actually went a little bit, we made our lives a little bit more difficult at the beginning by making it injectable, with the idea that chronic wounds are actually, they're not as superficial as normal wounds, right? If you think of ulcers, they can go actually pretty deep. So a wound dressing doesn't actually have contact with the whole wound, while injecting something that gels right at the side is a much better approach to have more contact. So we decided, let's start with that. You can always put that as a film on top of a wound dressing, right?
Yeah, that makes sense. Yeah, thanks. Is there any other questions? I like the bits about sort of talking to the future student. Future members of the lab. Very, very nice. All right, if there is not a burning urgent question at the moment, perhaps you can ask Nuria later in more casual setting. So thank you, Nuria. And next speaker is Juan Juan is, oh, let me find, I got muted, yes, and yes, all right. And Juan is a new lecturer in the department. You started back in, the long time ago, January 2020. And so before that, he was a postdoc in the Spanish National Research Council, CSIC, at Northwestern University. And you also did a PhD at CSIC and the University of Carlos III. You got PhD in 2013. Juan is multilingual. What are the languages you speak besides English and Spanish?
It's not that impressive. It's French and Portuguese. But
oh, that is pretty impressive. And Juan always put some Japanese into the email conversation I have with him, and it's always quite well, correct sort of Japanese. So definitely have a knack for language. He does say he has an accent in all those languages, including Spanish, and he also has an alter ego of artist, and I think you do creative writing and photography. Is that correct?
Creative writing is what I would love to do if I had time. I used to do a lot of photography back in the days.
Yeah, I'm not surprised hearing that. I mean, your website is so artistic. I really love it. With that, I am gonna shut up, and the floor is all yours, Juan.
Thank you, Naomi, for the super nice introduction. Can you see my slides?
Yes.
Oops. Okay. Yeah. Thank you very much. It's a pleasure for me to be here today. Yeah, I wish this could be in person, but I guess it is the way that it is. So basically, today I wanted to give you a flavor of the things that we're working, at the lab in general, and give you like a couple examples. And let's see if I manage to do this within 20 minutes or so. Like Nuria, I thought I could start by telling you a bit about myself, because I started like over a year ago, but this was like actually like 2 months before the pandemic started. So it's not, unfortunately, I haven't been seeing you as much as I would have liked to. But basically, I studied engineering. So I studied industrial engineering. So, I started like to basically build robots or factories or do like electric transmission or power transmission or electronics. But actually, along the way, I realized that I was more interested in understanding stuff. And the question that most caught my attention was understanding ourselves, meaning how we think and how we make decisions and why we behave the way we behave and why and how we see the world. So basically, to understand ourselves, I soon realized that we had to study the brain as the place from which the mind emerges. Although, I am one of those people who believes that in order to have a mind, you need your body, but this is a conversation probably to have some other day. So this is exactly what we do in the lab. We study the brain, and what we do to understand the brain is actually focus on the key function of the brain, which, if you had asked me 20 years ago, like what are brains for, I would have probably said like for thinking or, making decisions, but actually brains evolved to move. So basically in the lab, we try to understand the brain by studying behavior. And we know that brains were evolved to move because we have access to these polyps, these creatures that I call ascidia, that we think were among the first to have a brain. And actually what these things do is they are born and they kind of float in the ocean. And at some point they get to a rock that they like and they stick themselves to it, and then they digest their brain and remain there their entire life after having eaten their brain. And actually, Daniel Wolpert makes the analogy that ascidia are like full professors at universities. Once they get their chair, they can digest their own brain. I guess I shouldn't make that joke since I'm not a prof yet. But the point that these creatures help me make is that the brain evolved for moving, okay, which is one of the reasons why we study movement. And in fact, many of the most amazing things that humans do, I would argue, are related to movement, right? Like playing the guitar like at amazing speeds, creating beautiful paintings, or any extremely talented athlete that is like one of the most amazing feats. Being an amazing athlete is one of the most amazing feats that any human can achieve, right? Which is why we admire them so much. So, what does it take for us to move? So basically, moving actually requires using most of your brain, like almost everything. And if you imagine for a second that we are in a beer garden and having a nice conversation, I will use this to illustrate how this whole business works as far as we know, in a very simple way. So basically, imagine that I had my pint here. So at some point I would decide, I want a sip of my pint. And then that means that we'll have to identify where my pint is in the space. And, it has like this certain shape and it's at this distance. So, I know what I have to do. And then a collection of areas in the brain and the cortex and cerebellum orchestrate a plan that then gets converted into control signals that are sent from all these structures down to the spinal cord. And the spinal cord has the motor neurons that actually make your muscles contract and thereby causing the movement. But this is even more complicated than that, because if I'm reaching to my pint and you bump into my arm, normally I would be able to correct my movement in order to prevent the beer from spilling, unless I've had too many. And this is because there's like this continuous monitoring of what is going on in your movement that the brain uses to correct the movements very rapidly. Okay, so this is what we want to understand in the lab. And to do that, what we do is combine 4 ingredients. We study behavior, mostly in animals, but a little bit in humans as well. We record from many brain areas at the same time, because remember, this whole behavior is orchestrated by the coordinated activity of many areas. We also use analysis methods to analyze our behavioral and neural data. I guess we use machine learning using the jargon of these days. And we also build computational models that allow us to test and develop predictions about how things in the brain look the way they look. And what I want to do today is give you a flavor of these 4 aspects in something like 15 minutes or now. And I want to start by the first thing, which is the general philosophy of how we approach this problem. And the brain is made of neurons, actually billions of neurons, and these neurons talk to each other by sending small electric signals to each other. So based on knowing that, the neuroscience community traditionally sought to understand the brain by understanding the language that neurons in different areas speak. Okay, so basically, the idea was that if I know that neurons in this part of the brain care about these things of the external world, and these other neurons about this other thing, then I could basically write the dictionary of the brain and I would have understood the brain. Okay, and we call this single neuron coding because it assumes that neurons code for something. Okay, and the way to approach a neuroscientific question this way is basically to train animals to do a task. This is one of the tasks that I was doing in monkeys in Northwestern. So basically we had trained monkeys to move this yellow cursor by generating forces with their hand. And then the monkeys knew that they had to move the cursor to the target in order to get a little bit of juice. Okay, and they did this hundreds of times a day. And at the same time, we're recording neurons with electrodes implanted in their brains. Okay, and for example, this is the activity of one of the neurons we're recording from. And what I'm plotting here is how much the neuron is firing for its target. So there are 8 targets arranged as you can see here. And this is how the activity changes over time, if you follow my cursor. So basically this shows that after the target is presented, which is this dot, then monkey will start moving. So this neuron will fire more and then, a little bit less during the movement. Okay, and if you only saw this neuron in motor cortex, you will conclude that motor cortex cares about how you're moving your hand, because you see that this peak here changes its height depending on the target. Okay, so this neuron codes for the direction of the movement, if you want. The problem comes when you plot the activity of another neuron recorded simultaneously from the same brain area, only a few microns away from it, from this first one. And what you notice is that this neuron looks nothing like the first one. And if I plotted all 100 neurons that are recorded at the same time, they would all be different. Okay, so this already speaks about this idea of coding not being the whole picture. And actually things get even worse, because if you ask monkeys to do another task or to do the task with the hand in the different position, these curves will change completely in a way that we can't make sense of. Okay, so we think that we need another theory about how to understand the brain. And this is what the alternative that we've been trying to push along with some other colleagues for a few years now. This traditional idea that we have to understand what each single neuron does by looking at what the neuron is doing and how the monkey is doing, and what the monkey is doing, assumes that each of the neurons in the brain that I'm representing here as circles can change their activity independently to cause the movement. Okay, but we know that neurons in the brain make thousands of connections with other neurons, and we also know that this connectivity constrains what neurons can do. Okay, and the best way I can try to explain this to all of you guys is by thinking that you are, a neuron is like a person in a rock concert. When you are there, you are your own individual, so you can try to move somewhere, but if the entire crowd is pushing you in some direction, you won't have a lot of wiggle room to do something different. So in a way, we think that neurons are people in a rock concert. What matters is how the entire crowd is moving, and individuals are only like a small, like a sample of this whole crowd behavior. Okay. So here is our proposal. We propose that the brain doesn't do stuff or compute by adjusting independently the activity of each neuron, but rather what it does is adjust these population-wide patterns of neural activity that we call neural modes. Okay. And these neural modes are nothing more than the dominant patterns of activity that you record in the brain. Now, how can we test this in experiments? Because you can build a whole philosophy, but we need to test it and see if these ideas actually tell us something new. So imagine that now we're recording from these 3 neurons. Okay. Now what I want to do is study their coordinated activity, not what each of them is doing independently. So instead of plotting their activity over time, as I did before, what I'm going to do is represent the activity of the 3 neurons at the same time. Okay. And this means that at any given point in time, for example, when the monkey starts moving, their activity will be a point in this space. And as the monkey moves, this point will move, defining some trajectory. Plot all the trajectories for all the movements in this game that the monkey was playing. And then I can try to ask questions about how these trajectories look. And the first thing that I have to check, or that I check, is whether this activity is actually confined due to these constraints in the circuitry. And we can do that using mathematical methods to find this surface that we call neural manifold. And what we do find is that for all brain areas that we have checked, and across virtually all behaviors that people have checked, the activity actually lives in this lower dimensional structure, which means that actually neurons don't work independently, but work in a coordinated way. Okay, so what we do in the lab is trying to understand the properties of this geometric object, and the trajectory is defined by the activity to understand. And I want to give you a concrete example to illustrate one of the things we've learned. And we were trying to answer this very simple question, which is, how can we do the same thing on different days? Like, the typical thing we say that once you learn to ride a bike, you will never forget it in your life, or, musicians who learn to play an instrument and then, they may not practice for years, but then they catch their guitar again and they do it quickly, right? So we have this intuition that we don't forget motor skills, but how does the brain figure it out? So this is what my friend Matt and some other colleagues and me did. Basically, we had monkeys doing the same task on different days. They were not playing Ziggy Stardust, but doing a different motor task. And at the same time, we were recording activity from their brains using electrodes that were chronically implanted. However, even though the electrodes were chronically implanted, we were not seeing the same neurons on different days. This is something we know. These electrodes move a little bit inside the brain, so the neurons that you are sampling on different days may be different. But remember that we think that what matters is the collective behavior of all the neurons. So we thought that we should be able to find something stable in the brain as long as the monkeys are doing the same thing, even if the neurons we're recording from are different. Okay? And this is the hypothesis for this. So, if you are going to stay awake for 2 slides, is these 2 that are coming up next. So basically now imagine a space with d dimensions where these hundreds of millions, so each neuron in your motor cortex. Our idea is that when, for example, a monkey learns to reach this way with the robot, then the activity of these neurons will describe some trajectory, okay? That we call true latent dynamics, because it's the truth that we hypothesize causes the movement today, tomorrow, or years from now, okay? And this latent dynamics will live in some manifold. Now, I bring the monkey to the lab someday and I ask him to do this task. Well, I'll record from only these 3 neurons, okay? These 3 neurons will define some space, and we don't know the relationship with this first space. But we know that if our hypothesis is that when the monkey is moving, for example, this way, this trajectory should be a projection of this trajectory, okay? So in a way, if you want to think about it, and if we had a beamer, it would be as if you are, the true trajectory is my hand, and you were seeing a shadow in my hand, of my hand somewhere, okay? Now the trick is that you can record from 3 different neurons. You bring the monkey back some other day and you record maybe from 3 completely different neurons. But if you ask the monkey to do the same movement, you should see some other trajectory that will be again a reflection of a projection of this true trajectory, okay? So now, I don't know if you can see my hands well, but basically, if we are right and the activity of this population of neurons is the same when you do the same thing on different days, then what we think is going on is that on day 1, for example, you are seeing this hand, like my right hand like this, and in day 2 you may be seeing this. So the only thing that we need to do is basically rotate our hands to show that they are actually the same. Or in other words, we just have to find the right point of view to show that the blue trace and the green trace are the same, okay? So this is what we did. Basically, we had, one second about the task before I show you the results. We had monkeys using this joystick to move the cursor to a target. So basically, they were presented with a target. They waited a little bit until there was a beep, and then they moved to get a reward, okay? And this is just to show you that the behavior of the monkeys was very consistent. So this is the trajectory of the hands in from the top. So monkey reaching in front, toward the chest, and so forth. And these trajectories look the same over 2 years. But what I'm plotting here is the number of neurons that are the same on different days. So you can see that after 10 days or so, more than half of the neurons would have changed, okay? So actually we have very different neurons, but remember, we think this shouldn't be a problem, even if after a month there's basically no neurons that are the same as the first day. So here is the moment of truth. What I'm showing you here is what this population of neurons is doing when the monkey moves to each of these targets. So you can see, this is actual data, by the way. So you can see these trajectories that are color coded like the targets. All of them kind of start from this spot here and then do these circle trajectories. So this is for 1 day and this is 1 month later. And you can see that 1 month later, the trajectories look nothing like on the first day. And this is not surprising because we may be looking at completely different neurons. So it may be comparing my hand like this to this, right? Now the question is, can I find the right point of view to actually reveal that these 2 guys are the same? And actually we can. If you look at this, just by rotating these 2 sets of traces, we can uncover that they actually look very similar, okay? And we can measure that by computing correlations, and this is a correlation of 0.7, which is actually a very nice number. If you do neural data analysis or any analysis, we can compare that to how similar these trajectories are within 1 day when we're recording the same neurons, which is the gray dot. And we see that the activity of the brain is as similar across days as it is within a day, okay? And this is true over a month and a half. So this shows that actually the monkey may be doing the same task basically by using the same population patterns of neural activity that we can uncover even if we record from different neurons. And just to show you that we can do this over 2 years or 1.5 years in other monkeys. So this seems to be very stable. Now the last result I want to show you is what happens if this tells us something about how the monkey is actually moving. Because I've told you this is what is happening in the brain as monkeys move, but do these brain signals tell us anything about behavior? So what we did was take the activity of these neurons and try to predict how the monkey is moving the hand, so how the joystick is moving, okay? And we use very simple models to do that. And this is how things look. What I'm plotting here is the velocity of this robot on the horizontal and vertical axis. And you can see here that these traces look different. I should have said, sorry, that each of these traces is one trial, okay? So they look different because it's different targets, and the blank spaces separate the trials. So if I build a model on one day and test it on the same day, I can predict the movement very well from the brain, okay? Which is why the gray traces look so similar to the black traces. Now the important question is, can I build a model today and will it work weeks from now? And the answer actually is yes. We can build a model today and it will work perfectly well 2 weeks later, which is why the blue model is virtually as good as the gray model, okay? And what happens if I don't use this trick of aligning the population activity? Well, what happens is that my model basically sucks after 2 weeks. These green predictions, look nothing like how the monkey is moving, okay? And just to show you across, all the sessions, so this is how well we can predict in one day. And if we train a model today and test it a month and a half later, the model is as good as it can be. And if we don't do it, obviously the model becomes useless. And this is also true for 2 years of data and 1.5 years of data. Okay. So we think this is very cool for 2 reasons. One is because it supports this idea that the brain may use these population activity patterns basically to control movement. Another important thing is the application to brain-computer interfaces. So brain-computer interface is a device that allows a subject to control something with their mind. So this is a paralyzed volunteer who can't even move her eyes. So she has the same type of brain implant that our monkeys had. And Chetan and his colleagues basically use a model like the one I showed you before to predict how this lady wanted to move the cursor in this keyboard, okay? And this is an example of how this worked. So basically she's typing the lyrics to her favorite song, which is John Lennon's Imagine. And you can see that it's not as fast as typing on your keyboard, but it still is pretty good, overall, considering that she has been completely locked in for years. Okay. So this is all very cool, but this device has all of these devices have one challenge, which is this plot that I showed you before, that after a few weeks, the neurons you're recording from are different. So you have built your model on day 1 with some neurons, and then weeks later, the model is in completely different neurons, which means that it's not going to work as intended. If you want, like on day 1, your model is the perfect tool for the task, right? It's the beer opener to open your bottle of beer. But after 10 days, many of your neurons have changed, so you no longer have a beer opener. Now you have a coin. But the problem is that after a month or so, you have completely different neurons. So you basically have goggles to open a beer. So what our basic research shows is that we can take this BCI and instead of read the activity of the neurons as inputs, we can read this aligned latent dynamics, which means adding some math here. And with this, you will have a BCI that works in a robust way over years, okay? And actually, Alan Degenhardt and Will Bishop published a similar idea in Nature Biomedical Engineering a bit later than us, showing that it works online in monkeys, okay? And now to finish, I wanted to give you a flavor of the 4 ingredients of the lab. So I've talked so far about the behavioral experiments, neural recording, and data analysis. So I want to tell you a little bit about how we're using computational models of the brain to understand how it works. So this is work led by my student Jorge Benazar. Basically what Jorge did was build an artificial neural network model of the task I showed you before. So basically the network sees some input that represents the target that the monkey sees on the screen and is able to produce movements that have the same velocity profile as the monkey does. So basically the network learns to generate the movement that the monkey generates based on the targets, okay? And the super cool thing about this that has been shown like before is that this network, if you look at what these artificial neurons do, they look very similar to what the neurons in the brain are doing. So just to show you that, this is an example of one neuron in the brain, M1, and in the network, and the activity to 3 different targets. So reaching on top, reaching to your like 10 p.m. and to your 9 p.m. And you can see that the activity of this neuron is similar across repetitions for the same target. So yellow traces are together, orange and red, and they are separated across trials, both in the network and the brain, okay? And I can also plot these trajectories that I've been showing you for the network and the model, and they look qualitatively very similar. So we have a good model of the brain, and what we're trying to understand is how these collective patterns of activity emerge. So a way to look at this is to build these beautiful matrices that basically show how the neurons fire together. So the correlation across all the neurons. And we use some cluster algorithm to show this beautiful structure. So you can see that there are clusters of yellow here, which is what creates this correlation, of neurons being in a crowd in a rock concert all working together. Okay, this is data from a real monkey. And this is what we do if we simulate neurons that have more internal connections, okay? This first network has very few internal connections. This last one is fully connected. And what you can see is that, as we expected, as you make neurons that are more connected, like in the brain, you get activity that looks more similar to what the brain is doing. So we are extending this to explore more properties of this, of how this coordinated activity emerges in the brain. But I don't want to get into that in the interest of time. So in summary, I've been telling you about our research about how the brain works, basically, by looking at the collective activity of neural populations. I've shown you how we can use this idea to uncover basically how the brain does the same, how we do the same thing on different days. Among other things, we've used this idea to ask other questions, like how we do flexibly many different things, or how we can adapt our movements. I've also shown you how this work can enable brain-computer interfaces that work in a robust days for years. And I've given you a flavor of how we're using computational models to understand how this activity emerges in the brain. So to finish, I want to thank my former collaborators and to everyone in the lab for their amazing work. Again, like Nuria, I'm always happy to take enthusiastic students. So, you can email me, you can check our website. I'm also on Twitter, and also thanks to the guys giving us the money. And yeah, I'll be happy to take questions.
Thanks, Juan, for a beautiful, clear talk. And we have a question from Andre. Andre, do you want to ask a question yourself? Open up the microphone.
Yes, hello. Can you hear me?
Yes, loud and clear.
Yeah, so the question is, if you look at all the mappings from day 1 to day 2, or from day N to day M, basically every instance is when you have to turn one population to another. Right? Mm-hmm. Like in the simplest case, if it's a linear transformation, you can basically have a Jacobian that will do that job for you. Now, if you look at all those Jacobians, is there anything in common? In other words, like, do you have to just do something like that for all of them, or is it like, depending on the day, it's a totally different?
It's a great question, and it's actually a bit tricky to answer because these matrices live in different spaces. So it's like, you're comparing something that lives here with something that lives here. But I think what happens is pretty random because the way we think about it, and this is cooler if you do it in a room with a beamer, is like you have this object, like my hand, and each neuron is like a shadow of this hand. Like, you can actually, it's like a random shadow of the hand. So basically, depending on the neurons you are sampling, you are looking at this same thing from completely different points of view. Mm-hmm. Of course, if more neurons are stable, the rotations will be smaller, right? So we can play these games offline. Like we can drop like a small percentage of neurons or change a half of some of them, and show that we don't need to align a lot. And if we take two different, completely non-overlapping sets, we need to align more.
Yeah.
Yeah, but it's a very good question. I wish we could record the same neurons for super long periods of time to also understand it better.
Yeah.
But it seems to be very consistent across many brain areas, I mean.
Okay, thank you.
I hope that made sense.
Thanks, Andre. And Juan for the answer. Olivia, would you like to ask your question yourself?
Yes.
Or should, yeah, great.
Thanks
It's always.
That was really good.
Bonjour.
Again, so yeah, I was just wondering because obviously you've mentioned that you've worked with environments in the past, and you're working with mice, or you've worked with mice. And I was just wondering, like, working with two different species of monkey, two different, like, it's different challenge, I'm guessing, in ways. But in terms of tasks, like given a specific task to a monkey, two different challenges. And I was just wondering, like, what the type of task that you give, does that make it easier? Well, I guess there's different challenges working with mouse, trying with mouse, for example.
Yeah, it's a very good question. I think that you said the thing that is key to me, that is that we have to think that monkeys and mice and rats are all very different. So we have to think about designing tasks that are appropriate for the species, meaning that, you know, for example, we are going to use odors as cues for mice instead of making them look at ice cream, because mice have like amazing sense of scent. Like actually, there was a paper published in Nature last week, I think, showing that mice can scan odors at 40 hertz. So every 25 milliseconds, they can detect a difference in odor, which is much faster than we can see, basically. So this is one thing. And another thing is considering not only their senses, but also the way they behave. So for a mouse, it's very hard to sit still, right? And wait for a go cue. So basically, we're not going to enforce that in any task. We're going to let them explore more freely, because that is what mice like to do. And that will make some questions, you know, that will make some analysis harder, because you can train monkeys to sit there and wait for a cue and see what is happening in the brain as monkeys are thinking about what to do next. But there's ways around it. We think we are playing with some data from colleagues to find these signatures of planning actions and making decisions. Yeah, and I guess the other thing is the surgical techniques, but there's challenges in both, right? The mouse brain is smaller, but that also means that we can use the same electrode to recover from deeper structures, which is the way to monitor all these areas at the same time, which you cannot do in monkeys.
Thank you.
Yeah.
Great.
Thanks.
So the next question is from Chris. Chris, would you like to ask yourself? Or should I read your question? Are you there? So why was there a year gap? Oh yeah, okay, the microphone is broken. Right, there you go. Why was there a year gap between the monkey experiments? To see if they forget the task, or do you need to control for the other experiences the monkey have to see what affects the later forgetting?
Yeah, that's a very good question. So, you know, I had to go very fast because I wanted to show some data and also give you guys an overview in 20 minutes. So I hope it was, you know, I had to skip some details, but actually we had recordings every few weeks or months over these 2 years or 1 year and a half. So I can actually show that in a second, just so it's clear. One of these, so maybe this plot is clear. Like you can see that there's like, for example, this gap here, right, like of 100 days. And this is because for practical reasons, we're alternating monkeys between experiments just to be able to collect enough data. So someone else was using this monkey for different experiments in this time. But if you see, we have chunks of, you can see that we change monkeys like every month or something, right? There's like gaps. but we have like a lot of mapping over time. But you made your point is very good about forgetting. Like this is something that is very natural to them, right? Like once they understand the structure of the task, it's very easy for them to move a robot, right? Because it's very similar to reach into different directions. But when we're studying things like adapting to motor perturbations, like, we would add sometimes like forces that would push the arm away from the target, or we would rotate what they see on the screen. So the monkeys had to realize, oh, I'm moving this way, but the cursor is going this way. So for that, we did randomize and try to make sure that the monkeys forgot. But I don't think monkeys can forget this task because it's just like reaching to different targets. Yeah, I hope that answered it, Chris. Otherwise, just type on the chat. Oh, cool.
And we have somebody with raised hand. Yeah, Chris is happy. Okay, so I cannot see that. Okay, would you like to ask the question yourself? Oh, Masi?
Machi, I'm here.
Machi,
Hello.
yes, yeah, please.
Yes. So my question is: remember when you showed that you had the dimension space of the basically was a color-coded circle, and there were 8 arrows, I think, and after 32 days it was completely different, but you rotated it. How do you rotate it to match?
Yeah, this is a detail I skipped. So we use a method that is called canonical correlation analysis. That is actually very powerful and cool and general for any scientist. And the way CCA works is that you have one set of signals and another set of signals. And basically it finds the transformations that applied on these signals make them look as similar as they can be. So basically it means it rotates the signals and sometimes scales them to reveal that they are different. This is a very simple algebraic method that is based on matrix operations. But what I wanted to add is that we tried other methods to do this, like another thingy called procrastis analysis, even reduced rank regression, and also like fancy neural networks. And they'll give you qualitatively the same thing.
So standard image processing techniques.
Well, CCA is an everything processing technique, but yeah, nothing fancy. So we like using simple methods and trying a few to see if we get the same effect. Because if one method gives you a result and another kind of similar method doesn't, then you have to be a bit suspicious, I think, with this kind of data.
Okay, thank
you. Okay, so thank you for the question and answer. I think that's the end of the pouring in questions. I think we should stop the official part of the seminars, and both the speakers can stick around a little bit to have a more casual Q&A session, so please stay around. So now we're going to stop the recording with the official